I'm going to use the old-fashioned communication medium of talking to you. I'm actually very happy to follow Ben because I think about different modes of communication a lot, and I actually like to reference not just Web 3.0, Web 2.0, but Web 1.0, the medium originally known as hypertext, which I think is woefully underused today. Typically, when you see links these days, they're either references to external works, they're navigation links to help you find something on a site, or they're part of a wiki. I think that the concept of hypertext has almost been folded into wikis and collaboration, and the idea of an authorial work of hypertext, where you're taking a dense network of concepts and linking them to each other, has sort of gone by the wayside. That's actually how I'm trying to present my project. So I should say a little bit about what I do. I often describe my project as trying to understand the simplest brain in nature, but I realize that it sounds cooler and also it's somewhat more accurate to simply say that I'm building a robot that uses lasers to do mind control. It's actually is closer to the truth. And of course, I'm working with a very specific type of mind, the mind of a nematode worm, which is transparent and genetically engineered. So don't worry, your minds are not in danger of being controlled anytime soon. But the robot and the lasers is totally accurate. I'm trying to do these experiments in an automated fashion and completely optically, which is something that, 8 years ago would have been rightfully considered pure science fiction. And so the amount of technology that we have now that we didn't not so long ago is really staggering. And my goal in presenting my work is to try and communicate the awe that I have of our actual capabilities, as opposed to trying to make these sort of general sweeping claims that gloss over the details. And I was particularly peeved by this headline that came out recently that says Canadian scientists create first functioning virtual brain. When the actual thing that they did, which is very cool, is that they created a neural network that can solve more than one task, whereas previously neural nets could only be trained to do one thing at a time. And neural nets are experiencing this really interesting renaissance, where for a long time it was considered neural nets were a dead end. They weren't really useful for anything. They certainly didn't resemble actual nervous systems. And now we have deep belief nets, and we have these interesting systems like Spawn, the one that headline was about, where you can organize neural nets to share different pieces so that they can accomplish different tasks, but also exchange their learnings with each other. So there's a lot of interesting things happening in neural nets, but instead everyone writes about it as though we've solved the brain now. And so I want to talk about some of the really fascinating technologies that have recently come out, like two-photon digital holography. And I just think it's crazy that there is an actual thing that happens that's special when two laser beams meet each other. It is a sort of this fantasy that you have, where like, oh, when two lasers cross, it's like lightsabers. And something special happens in the middle. And actually, if you take two lasers, generally nothing interesting happens. It's quite disappointing. But if the lasers are sufficiently fast, if you're using femtosecond pulses, then the amount of photon flux at the point where the beams meet can actually reach a sufficient level to cause these two-photon events, where two photons from either side actually arrive at a single molecule at a single instant, so that the electron does not have time to decay after encountering the first photon before the second photon shows up, and it's as if a single photon of twice the energy had arrived. And that doesn't happen anywhere in the surrounding space. It only happens where the photon flux crosses this two-photon threshold, and so you can use that to cause effects in a three-dimensional sample where they only occur at a single point or a single set of points, and not in the surrounding neighborhoods. And so this is a critical technology for stimulating an individual neuron by computer control in a three-dimensional nervous system, as all living nervous systems are. We're not talking about slices. So that's a cool thing. It's a cool thing that we have like InfiniBand RDMA, which can combine individual computers into a supercomputer in a way that we couldn't before. And that sounds very hyperbolic, because we have supercomputers. And of course, we've had supercomputers for a long time, and you can always network computers and you can do distributed computing. But the cool thing is it's reduced the latency. So you no longer have to do an act of communication. Actually, it kind of goes back to what Ben was saying, where you take your data packet, you format it into internet protocol, and exchange it across a network, and then it's received at the other end. Now with InfiniBand, you can have RDMA means remote direct memory access. So one computer can directly access the memory in a remote computer. We couldn't do that before. That's really cool. And that enables a new class of applications, very high throughput image processing, for instance, that are necessary for what I'm doing. There's also we have good jitting compilers now. This just crossed the threshold a few weeks ago, and it sort of made quiet news in the computer science community. Showed up at maybe number 20 on the front page of Hacker News, and that was about all there was to it. People talk about self-modifying code in Lisp, for example. But the Lisp macro system takes place when you're actually compiling the code. So in a single step, after you've written down your expression, the compiler can run your code on itself, translate it into a form that is in more detail, so that you can be more poetic and expressive in your Lisp coding. This is extremely powerful, and people rightfully talk about how great Lisp is, because you can do this sort of macro coding. But once you've done it, once the program is running, all you have is eval, which is cumbersome and slow. You can create code and you can run it, but it costs a huge amount of overhead. And so it's not really self-modifying code in the sense that as it experiences the world, it can adapt to changing circumstances. It's only self-modifying in the sense that you're writing a program to write the program for you. But we've actually gone through that limit now. LuaJIT 2 is the name of the thing. And I think this is really important. It's now, with almost zero overhead, you can generate code on the fly that runs at native speeds. So that's another thing that I'm using in my machine learning system. So there's all these really cool things, and I'm just trying to express that. And the way I'm doing that is through hypertext. So I have a web page for Nemaload, where I give a very short overview of the project, but all of the fancy words like transgenics and 2-photon and holography are all hyperlinks, and then I try to explain each of these new technologies and how they work with each other, and how they can be used to produce this one single result that draws on a number of different fields. So basically, my message is when we're writing about the future, we shouldn't be scared to be concrete about the actual capabilities that we have and what the actual factors are. What are the limiting factors that are removed by each new innovation? What are the new things that we can do that are not just interesting steps towards some abstract future, but actual capabilities that we now can take into our hands and apply to problems now. Like understanding the simplest brain, which, I mean, understanding how the nematode thinks. It's 302 neurons. It's very simple. Its most interesting behavior is that it likes to be at the same temperature that it was raised at. Okay, this is not a very smart creature, but it's still miraculous that 302 neurons can produce all the behaviors that it does. It's sensitive to a number of odorants. It locomotes. It can navigate to whatever kind of temperature or chemical concentration it wants. It's sensitive to light. It's sensitive to touch. It turns around if you tap it on the head. It does all of these things, and we don't understand how it works. And nature has organized these computations in some way. And so we don't even need to talk about cortex to talk about the mysteries of brain computation. Even the very simplest brains that have nothing resembling cortex still hold interesting secrets, and we're reaching the point where we can begin to unlock those, where we can begin to look at what's going on. The big quote that I have on the front of my website is from Richard Feynman, and he was talking about the electron microscope, which came to fruition during the course of his career. And he said, in his talk, there's plenty of room at the bottom, he said, all of these fundamental biological questions are going to be very easy to answer when you can just look at the thing. And new tools for looking at things are really important. You can then look at them and see how they work. Then you can use those insights to build your own copies, your own versions of those systems. So I can say a little bit more about also my background, how I came to be working on this project, because another big part of my mission in doing this is to show that there are different ways to pursue research. So I was sort of originally an artificial intelligence student at the MIT Center for Bits and Atoms. And I was working on a computing architecture which is based on cellular automata, which is a really horrible idea. If anyone is thinking about cellular automata computing, just don't go there. It's just not very practical. It's fun to think about. And so there's this big project, this vertically integrated thing of, all right, we're going to create a programming language for your cellular automata, and then we're going to make this modular AI system on top of that programming language, and so on and so forth. And I took a break from that to go to Singularity University. And the very first day of that, Larry Page shows up as a surprise, and someone asks, "What do you think about AI?" And I'm like, I work on AI. I should pay close attention to this. And he says, well, I think there's two ways that we can go. Not enough smart people are really working on AI. The first is you just use massive scale, and you go through all of the algorithms until you find the algorithm for intelligence. And this, I think, is based on a fundamental misconception that Larry has about how much computing power there is in the world, because I suspect that he can't fully internalize the fact that he has most of it. So he's probably thinking, you know, I have a Google, there's probably, you know, a million other Googles. If we all worked together, we could, yeah, but it's not going to work quite yet. We don't actually have that much computing power. But then he said the second way is if we start by just understanding the simplest brain that's out there in nature. And we get that really nailed down, and then we start to work our way up the evolutionary ladder until we understand a brain that has some intelligence. And then, ta-da, we understand intelligence by extension. So I started to look into, you know, what's the reason that we aren't doing this? Why haven't the biologists figured out this 302 neuron thing? And so I downloaded the connectome. There's this whole thing about connectomes. If I had slides, I would have shown you Sebastian Sung's face rendered as a neural network on the cover of his book, Connectome. And he's making this whole argument, which overall I'm very sympathetic to, which is that your identity is tied to the state of your brain, rather than anything more mysterious. But he's being particularly restrictive about it. He's talking strictly about the anatomical state of the brain, about which neurons connect to which other neurons and where the synapses are. And there's these projects that people are starting to collect the connectomes of, for instance, an entire mouse, which, given about 5 years of tool development in electron microscopy and automated slicing, so you have to do these things in slice, would take about a year of literally slicing and imaging continuously to produce the data for. And the result of this would be you'd have a map of where all the connections are in a mouse brain, but we already have a map of where all the connections are in C. elegans. And C. elegans is significantly simpler, and it doesn't tell us anything. Really? And so at first I was skeptical of this. I was like, well, no, clearly no one's tried. Like, we have the connectome, right? There's a complete architecture. So I downloaded the connectome, and I started writing a program to simulate it. I was like, all right, so we have 302 nodes, and here are all the connections. So I'm going to create connection objects between each of the neurons that are connected to each other. And now each of the neurons has a state. So I'll just assume the states start at 0. And then for each connection, then I realized we have no idea what the connections do. We only know where they are. So this is a constraint. You know, it's a constraint on the model. It says these things which are not connected, they probably don't influence each other, although in fact they probably do through long-range peptide diffusion and so on, but at least it tells us most of the important things are probably happening between these sets of nodes. It gives us a graph, but it tells us nothing about what those important things are. And so in order to learn that, we actually need to look at what the animal is doing while it's living, not just when it's dead, where are the things, but what do the things do? And so in order to do that, we need imaging technologies that work in living animals. And that's something that actually hasn't been possible, surprisingly, until quite recently. To be able to look at what's going on in a living animal requires not just the optics, not just the computation, it also requires synthetic biology, which incidentally, I discovered a really great definition for recently. The definition is making DNA from scratch. So you're not just taking a gene from some other organism and splicing it in, but you can make whatever it is that you want. And we can use that, and scientists are using that now to do things like create neurons that glow optically in proportion to their activity level so that you can look at what they are doing. So these are the things that I'm working on. I hope I have amused and informed you. And now it's time for the next speaker.
Will we have time for
questions?
Yes.
How many synaptic bonds does 302 neurons create?
So the number of synapses is not quite as reliable. It's always 302 neurons. In the one animal that they did an extremely detailed map of, there are 7,283 synapses, but that's give or take 100. Yes?
At Singularity Summit a month and a half ago, Melanie Mitchell talked about cognition being based upon analogy. Mm-hmm. And when you were talking about hypertext there, it seemed to be very similar. I wondered if you want to talk about the connection between those two ideas.
Sure. Yeah, so I think one way that in my mind I reified Ben's statement about sort of communication being a mind is that I imagined that mind as being sort of an autonomous entity, and then the first step in the process of communicating is you put your mind into resonance with that mind. And so through learning, it mirrors the structure that you have in your mind that you wanted to communicate. And so that creates an isomorphism or an analogy between that structure and the one you're trying to communicate. Then once you're satisfied with the degree of similarity, then you send that little package off, and then it puts itself into resonance with a recipient mind, and it creates an isomorphism there. And so I think when we're talking about hypertext, it is kind of like that. When we create a collection of things that are linked together, it's not quite the same, because when you experience hypertext, you're not experiencing it as a graph. You still are experiencing it as one thing at a time, and so you get these things in sequence. But you get them in a sequence that makes sense to you. You're not getting them in whatever sequence the author decided was most pedagogically favorable. You're guiding that process, and so in some sense, it was in a very restricted way. It's adapting itself to your mind, as opposed to the way that it's serialized by the sender's mind. But we could do much better than that. One of the things that I'm working on is to actually put some code into this, and that's where it gets into the Web 2.0 stuff, that tries to estimate just a few numbers about, okay, how much does this user know about biology? How much do they know about math? How much do they know about computer science? And then to adjust the level of the terminology that's used accordingly. Yes?
That was a heck of a talk. Thank you very much. This business of outside stuff coming into a given field, like this lasers coming into this, is really what it's all about in terms of things happening. Yes. And that's, for example, the high efficiency lasers coming in is what I was talking about yesterday for getting into space. Mm-hmm, mm-hmm. So yeah, the great things happen when previously separate intellectual concepts combine. And one of my big problems with the standard academic institution is the degree of separation that's placed between, and it doesn't even matter between what and what, just how much separation there is between anything and anything. It's inexcusable. I mean, yeah.
Hey David, nice to see you. Great talk. So I don't know if you've read Ray Kurzweil's new book, How to Create a Mind, but
skimmed it.
Your research with the C. elegans, consistent with Kurzweil's new theory of pattern recognition and theory of intelligence?
Well, Kurzweil's theory is based on observations of the neocortex, and as I mentioned, C. elegans doesn't have one of those. So my research can't really support or disprove what he's doing. He actually does have a paragraph on me in there, and he sort of says the same thing. He says, well, there's this guy, Dalrymple, he's doing C. elegans, but it doesn't have a cortex, so it's not really relevant.
Thank you very much.
Yeah.
Our next speaker is Hermes Nam, who's a well-known author of, he started out with science fiction